Oh, sorry, I was muted. Thank you for the introduction. Can everyone see my screen?
Yes.
Okay, great. So my name is Michael Fanous. I'm part of the Quantitative Light Imaging Lab here at Beckman under the direction of Professor Popescu. And today I'll be talking about phase imaging with computational specificity, also known as PICS, principle and applications in biomedicine. Here, I'm just going to be covering a small portion of the projects that I've been involved with using these techniques. If you find any of these results exciting or compelling and you want to look into more, I really encourage you to check out our webpage and to look at the various articles that we do using these techniques. So first, let me introduce the topic. PICS, what is it? Basically, I'm sorry? Basically, what is PICS? PICS is a combination of QPI, quantitative phase imaging, with the power, efficiency, and speed of AI computation. So the reason why we use quantitative phase imaging in the first place is because it circumvents a lot of problems that fluorescent modalities have, such as photobleaching, phototoxicity. We don't need any of this photo damage because we don't label our cells. We can look at unlabeled cells. We can look at it with high specificity, and we get the added bonus of quantitative information, which is associated with the optical path length and therefore dry mass. So that allows us to monitor growth, in live real-time dynamics with cells. Here's an image that's taken from the original paper PICS published last year, and it's showing you here on the left. Let me get my laser pointer. This is the system that we use, and it's very convenient because we can take both quantitative images and fluorescence back to back, and the field of view is perfectly registered. So this kind of data is very conducive to have models trained to predict the fluorescence from the phase. So the ultimate goal here is to get rid of the fluorescence, to predict it using the model, and only having to image label-free data. Here's an example from that paper using human colon adenocarcinoma cells, and these cells have nuclei. The nuclei is stained with DAPI, and on the right here you see the ground truth image, and that's showing you the nuclei. And here we have PICS, so this is a prediction. So what we would do is we would apply this then after the fact to cells that have not been labeled, and therefore we get the advantages of the fluorescent specificity to delineate components without the tags, without the photo damage, plus we get the added bonus of the quantitative information, which allows us to monitor things like growth. Now in this case, in this talk, I'm going to cover two types of projects, two different classes of projects. One is pathology projects, which lend themselves well to potentially be employed in a clinical setting. And then the second type would be like a basic scientific analysis. In our case, looking at real-time myelin production in oligodendrocytes and their interactions with neurons. So the pathology projects, very briefly, one is myelin. I've been working on different samples of myelin, assessing myelin in brain tissue, piglet tissues. And we've recently introduced AI to that, and we have some extraordinary results for classification tasks. Another project is white blood cell counts. So we have a number of blood smears, and we want to look at the white blood cells very quickly, identify them, segment them, classify them, etc. And currently I'm working on breast biopsies to look at collagen signatures associated with the cancerous features, and that's done in collaboration with Carl. So let's start with the myelin. All right, why are we interested in myelin? What's the point? What's the purpose? What's the motivation? Well, myelin deficiencies, unfortunately, are related with serious cognitive difficulties such as MS, Alzheimer's, temporal lobe epilepsy. The myelin that surrounds the axon, that the myelin sheaths, are obviously vital for the nerve conduction, or impulse conductions. And it's a key component in evaluating the cognitive deficiencies that plague certain child developments. So we're particularly interested in the internal capsule of the brain. So the internal capsule here would be the area that goes to and from the cerebral cortex, carrying the information. If this is compromised in terms of myelin, it has serious consequences. So we were presented, and this is in collaboration with Abbott Labs and also researchers from the Department of Science here, Animal Sciences, and we were presented with 19 brain pathology slides. Now these were taken from piglets who were previously measured before they were euthanized with MRI. Now, MRI has a number of drawbacks. It's a phenomenal modality, but it has certain shortcomings, notably the resolution and the specificity. So it's very hard to accurately assess the degree, the amount of distribution of myelin in these brains looking from MRI. So what they did was they presented us with these 19 slides, these coronal sections of brain tissue, and they said, okay, just go and analyze it, try to find the myelin, and tell us what you think. We were blinded to what the categories were. They were actually divided into 4 categories. Something called gestational age, which is really the size of the specimen when it's born. And if it's appropriate versus small, the appropriate is likely to have more myelin. It's just more developed. It's a healthier specimen. The other thing that they did was they divided the piglets into 2 groups, one with an experimental diet and the other without. And the experimental diet included hydrolyzed fats, which is associated, they think, with an increase in myelin abundance. What we did is we imaged this with something called CSLIM. You may have heard of spatial light interference microscopy. Basically, we take 4 interferograms. This is using a spatial light modulator, and we're able to extract pure phase information. In this case, we swap the objective with a bright field objective, and we also use a spatial light modulator to be able to shift the unscattered light 4 times. It's a big technical, but basically the idea is we're able to extract pure phase information. Now, that information is associated with the optical path length, which is associated with dry mass. The idea here, and the beautiful thing, is that the first frame taken with an RGB camera is your basic bright field micrograph, okay? So here you can see this is a frame of piglet tissue that was stained with LFB, Luxelfast blue, which allows you to look at myelin in blue, and everything else will be in pink and purple. So here's the segmentation scheme that we did. Keep in mind, this has no AI. I'm just trying to set the backstory here so that you can understand the idea. We have corresponding bright field and phase images. Now, why do we use this? Because we want to get all of the myelin information rather than the cells and the dendrites and all of that, which is not pertinent to really evaluate the myelin distribution. What we do is we take this, we threshold it, we label 1 everything that's blue and black everything that's not. We just multiply them, a very simple multiplication, and then we get pure phase of the myelin, okay? Then we add all these together and we look at the statistics. Then Abbott Labs comes to us and tells us the answers. They say, okay, this slide had AGA versus SGA. This slide is associated with piglets before they were euthanized. They had a certain diet regimen. So they had high hydrolyzed fats. And what we found statistically significantly was that hydrolyzed fat actually increased the abundance of myelin. Very good, a successful study. We published it last year. Then we had some ambitious questions because we were starting to introduce AI into our computation analysis. This was done in collaboration with Dr. Saab from NSCA, and he had a few really intriguing ideas, some ambitious questions that we wanted to address. And could we do it? So we took all of our data, it's about 10,000 slides. We divided it into 4 categories, size and diet. And you did your basic 80%, 10%, 10% test validation. And then these were the questions that we wanted to ask. Okay, so keep in mind, this is kind of unique. Given 1 frame of a quantitative image of this piglet tissue, can you tell me that the piglet that's associated with this, was it appropriate for gestational age or small? Secondly, was it fed a diet of hydrolyzed fats or was it control? And thirdly, can you do both? Okay, so this is what the diet looks like. On the left, you have here the Luxelfast blue. This is the whole brain section. We take a very small portion of the internal capsule. Again, the internal capsule is especially relevant for cognitive deficiencies involving a dearth of myelin. Okay, so this is what it looks like. This is a scan. It's about 25 by 25. We're talking about 650 images. Approximately, we have 19 slides that amounts to about 10,000 images. So the degree of data wasn't a problem. Here's the problem. The problem is all of these phase images look more or less the same. A histopathologist, an expert who knows these slides, would not be able even remotely to discern the differences from these categories based on these images. So we're thinking, okay, it's got to be a bit more complex than simple numerical analysis. What we did was we just took the histogram of all of these numbers. So this is quantitative data. The intensity values have meaning. So what we did is we just paired them. We looked at the contrast between different groups and we said, okay, is there any prominent discrepancies? And this is a graph that's basically displaying no. So it's not that simple. You can't have a basic manually done algorithm to differentiate these two things. So then we started experimenting with transfer learning networks. Transfer learning networks allows you to classify data based on a label. And we hit upon something called exception. Exception just happened to be the best one. We were also doing MobileNet too, and then various things like that if you're familiar with it. Basically, the idea is you put it through this neural network, and finally you're able to answer certain questions. Is the frame associated with diet, associated with an age, unassociated with both? These are the results. Basically what this is saying, this is accuracy and loss blocks. So basically what this is saying here is that this is the hydrolyzed fats bit. So it's saying that with an 80% accuracy, I can take one frame, one quantitative frame from one slide, from one internal capsule region, and I can tell you with 80% certainty that that is associated with a piglet that was fed hydrolyzed fats. Now that's kind of an astonishing result, even if it's 80%. You know, a lot of these medical tasks that look at like glands and cancerous things. These are things that a histopathologist could know directly, very immediately. And that's where the label comes from. The label here doesn't come from that. It comes from the circumstance of piglets where they were euthanized. So that's unique. Now, if we look at all four categories, which is extra ambitious, those numbers go down quite a bit. So you're about 65%, which is not necessarily as impressive, but still keeping in mind what the overall goal here is, I think it's fairly astonishing. Now, here's one problem. We have all these 16 slides. We divide them into training and validation and test, and the tests have never been seen by the model, which is great. However, you have slides, right? The slides have certain unique features, which may have nothing to do with the tissue itself. It may be the way that the slide was made, the way that the thickness might be different and so on. So the model isn't trained to know that. It's just picking up on features and patterns that allow it to guide it to the right solution. So this is a slide that we hadn't introduced to the model whatsoever, not training, not validation, not test. And this happened to be one associated with a piglet before it was euthanized, fed an experimental diet of hydrolyzed fats, and was small for gestational age. The results were pretty good. They were actually fairly encouraging. So if you look here, SGA, control SGA, diet, this is a confusion matrix. You just add these two together and you get an accuracy of 94%, fairly good. If you do the diet, it's slightly less, it's about 77%, but still interesting. And as far as we're concerned, this is a tremendous success because again, this is not even remotely possible for a histopathologist of the first rate to achieve. Okay, now let's make a little shift into something which is a little bit more, foundational in terms of science. We want to look at how are these neurons even being myelinated. In case you don't know, the neuron, which has an axon, which needs to be myelated in order to conduct nerve impulses, needs myelin so that it doesn't lose the electricity of the impulse. Now that myelin doesn't come prepackaged, okay? It needs to interact with something called an oligodendrocyte. So here are some oligodendrocytes. They look fairly astral in their configurations. They have a lot of dendrites coming out of them. And here they are in a cold culture. They're dynamic. They're interacting with a neuron. They sense the neuron here, and you can see that they're reaching out to this axon and trying to coat it with myelin. So this is really cool. This is unprecedented as far as we're concerned in terms of evaluating the growth of myelin on an axon directly. And the reason why we're able to do this is because biologically, the axon is not supposed to increase in diameter all on its own. By the way, keep in mind this is unlabeled. So we don't know where the myelin is exactly. But
Michael,
yes.
You only have like 1 minute left, so just
Really?
for you to know. Yes.
Okay, let me wrap this up. So in collaboration with Dr. Andrew Steelman, what we did was we labeled these with PLP and F and NF, which is a stain which allows you to find the axon, and the PLP is the myelin. And so we predict this in a similar way with the colon cells. However, this is not as nice and tidy as a nucleus. So it's a little bit more involved, and we're still fine-tuning that. All right, very quickly, I want to go over a different project, a pathology project involving white blood cells. Now, the white blood cell, the reason why we're doing this is because usually it involves flow cytometry, which can be cumbersome, time-consuming, and not thoroughly practical. What we want to do is maybe take a finger prick, put it over a slide, make a blood smear, and then figure out all of the leukocytes and what they are, where they are, and what is the degree of their and how healthy they are. So here we're doing CSLIM again. We got these, we have counterpart images. So we feed them into the algorithm again, and we're using something called GAN. GAN is the generative adversarial network, which basically means that you have a generative portion and a discriminator. The generator's task is to produce fake images and fool the discriminator. They're working back to back in a zero-sum type game. And once they reach about 50% success, that's it, it's converged. So you may have seen this kind of thing where you take a picture and then you produce a painting. We're doing the exact same thing. We want SLIM to produce the bright field image and bypass the need to use a stain. And actually, it's a fairly good result. We're using the PICS to PICS model. And we're looking at 4 different types of white blood cells. We classify them both in localization and pixel values. And this was done also in collaboration with the Professor Anastasio's group.
Michael, I'm really sorry. I really have to cut you here.
Off?
Yes,
okay.
I'm sorry. I don't like doing that, but we have 2 other presenters.
That's fine. This is
I would rather have you have some question rather than like not having anything else.
No problem.
So, okay.
I hope I
So
wasn't too fast, and some people grasped some of what I had to say. There's a lot more results from this. I tried to trim it, but couldn't part with certain slides, unfortunately.
It's fine. Does anybody have a question for Michael? I have a quick one, and then we can like follow up. So you talk about like classification of cells in terms of size, mainly, and I would like to ask like more, like, so now you are able to recognize cells, but is it possible to recognize with your process, to recognize some process instead of a cell, like something that is moving, some process occurring inside the brain, for example? Do you think it's possible?
Well, this is not dynamic. If
it is static. Okay.
Yeah.
Okay. And is it something that could be interesting to do that in dynamic rather than static?
Well, to do it in dynamic, you would need to do a co-culture. So you couldn't get a biopsy or something like that. It's always complicated getting tissue from the brain when the person is living. You need to go in and drill a hole through the cranium. So that's not optimal. And you have to keep in mind that these diseases like multiple sclerosis, AD, they're very hard to diagnose. I mean, the symptoms of multiple sclerosis are all over the place, and people think they have it, people don't think they have it, they might have it, and so on. So this is a way to this is a last step, okay? You drill the hole through the brain, you get a piece of tissue, and then you stain it with us, and we give you an 80% accuracy. So that could be, fairly valuable.
I got it. Okay, thank you.
Hi, may I ask a question here?
Sure.
Of course.
So since you haven't gotten a whole lot of time introducing your data set, can I ask, is that a 2D data set or 3D one?
Which data set?
Mostly the axons. Yes.
That's all 2D.
Okay. So it's not stacked?
No, this is a tissue slide that we scanned.
Yeah, I see.
It's all 2D. There are no Z stacking of any kind. But we have 2 modalities. We have bright field, and then we have the phase image. But yeah, we didn't know. These are very thin tissue slides. There was no need for that.
Perfect. Thank you.
Sure.
Okay, thank you. So we'll move on to the next presenters. So I'm glad to present Nil Parikh. He's a PhD student in aerospace engineering, working with Prof. Gobet and Prof. Sotos. And his research encompasses like bio-inspired manufacturing and digital design. And this is for application to mechanical and structural composites. So Nil, the floor is yours now.
Hello, everyone. As Julie introduced me, my name is Nil Parikh. I'm a fifth-year grad student in the aerospace engineering department, and today I'll be presenting on my work towards 3D printing of bio-inspired fiber-reinforced composites. So the project itself is a combination of 3 domains, and we'll take a brief look at all 3 of them today and how we combine them in order to facilitate and realize the 3D printing of fiber-reinforced composites. So first, we'll take a look at our inspiration as well as our motivation for this work, and we'll take a look at natural composites and the architectures present within them and how we can translate that to manufacturing and synthetic analogs. And then we'll look at the materials that we're going to be utilizing in order to print these architectures, and then we'll end the presentation with the printing mechanism that we're designing as well as developing in order to facilitate the fabrication and printing of continuous fiber reinforced composites. So to jump in, not everyone is familiar with this, but what is 3D printing? It starts with a digital design that you can make a part within a computer-aided design software, so a CAD software, and then that is fed into a slicing software, which, as the name suggests, takes your part, cuts it into pieces, and then allows the motion gantry and the software to create a G-code that's used in order to facilitate the printing. And so the gantry moves around and allows fabrication of the part. So once that's created, there's a large variety of 3D printing technologies that you could adopt in order to actually print this. But our focus is going to be primarily towards material extrusion as well as direct energy deposition for this talk. And so the other part of this talk was about composite manufacturing. So just taking a quick look at how traditional composites are manufactured. For those of you who are unaware, a composite is a combination of 2 or more distinct materials in which we reap the benefits of both each of the constituents for a tailored performance of the material system. So composites are typically used in a wide variety of applications. So here we see the Boeing 787 up at the top. So the aerospace industry, it's used in the automotive industry as well as the energy industry, to name a few. Traditionally, these are manufactured in these large molds. So on the top is the mold that's used for a wind turbine blade, and it's typically heated up in order to cure the part. On the bottom, we have an autoclave that is used to cure the parts for the wings as well as the fuselage for the 787. That's shown in the upper left-hand corner. And then on the right, we'll see a representative curve that's typically used to cure these parts. And as we can see, this goes to elevated temperatures as well as pressures. And so regardless of the advances and the benefits of using these types of materials, the challenges associated with this include the long cure times necessary to cure the material itself, the high energy consumption, as well as the large capital investment for these types of tooling, as well as the parts itself. And so looking back, and to alleviate some of these manufacturing pitfalls, we look towards nature's manufacturing schemes. So natural composites typically employ elaborate hierarchical structures in order to achieve outstanding function, as well as mechanical properties with the materials that are readily available around them. And so one such structure that we'll be focusing on is the Bouligand type structure, which is consists of a uniaxially aligned and layered rotated microstructure that's given in sheets. And so this imparts impact resistance as well as deflection of cracks through the rotated and layered microstructure. And you can find these in the dactyl clubs of the mantis shrimp, the scales of the Apernagigas, the tusks of the African elephant, as well as the brick and mortar structure that's found typically in nacre. Another type of structure that we're going to be looking at is the cellular microstructure, and that's primarily seen in the trabecular bone, the shell of your average sea turtle, the glass sea sponge, and the wings of the blue morpho butterfly. And so the main advantages of this are reducing the amount of weight while also retaining its compressive and impact loading conditions. And in the case of the blue morpho butterfly, it can optically transform its properties based on that structure. So for our work in collaboration with the Gubel Group, as well as the Tissue Biomechanics Lab, we're going to be coupling computational imaging of these microstructures that are present within these natural structures, as well as mechanical modeling in order to design the structures that we're going to be then using in order to create the G codes for printing. However, we do have to look at a new curing scheme in order to actually realize this 3D printing. And in order to enable this rapid curing, a method that's garnered significant interest is frontal polymerization. So this reaction occurs when heat is introduced to a latent catalyst, which activates it and results in an exothermic polymerization, which releases subsequent heat. And that cycle continues on and on until all the catalyst has been exhausted. So this technique's been applied to a few different composite applications that you can see on the bottom, and I won't get into great detail with these. However, we can see that, yes, in fact, you could make a composite with this. However, it does have the rapid curing that we need. It has the minimal amount of energy, which alleviates some of the pitfalls that we saw previously in the composites manufacturing. However, it lacks in terms of high performance applications, as well as the complexity of the parts that we can fabricate. And so this really drives us towards integrating this new technique that the Autonomous Material Systems group here at the Beckman Institute's been pioneering as well as pushing, is to use this dicyclopentadiene monomer in the presence of a ruthenium Grubbs catalyst and alkali phosphite inhibitor. And that's introduced to a thermal stimulus, which converts the monomer into a thermosetting polymer. So this polydicyclopentadiene. And this process is actually quite interesting and well visualized in a test tube video shown on the right. So a thermal stimulus is applied on the left end by a soldering iron, and you can see the conversion of monomer, which is the pinkish color, to the polymer, which is the yellowish hue color that you see here. And so the process has a high enthalpy of polymerization, and it results in a high performance thermoset. And as I've previously shown, you can use it to fabricate these composite parts. And this was for a fiber reinforced composite of around 51%. That being said, to integrate this into 3D printing, we have to look at the integration of the resin system within the fibers as we're depositing the material in 3D printing. So in order to do that, we're going to be taking advantage of the liquid absorption coefficient, looking at the properties for the resin system. So here, the density, the viscosity, as well as the surface tension. The fiber properties in terms of the porosity, as well as the tortuosity, and then the interaction between the fiber and the resin itself, looking at the contact angle. And so the idea here is to optimize the liquid absorption coefficient. So I plot this on the bottom as a function of temperature. One thing to account here is you want to retain the viscosity and the resin properties over time. And so we want to minimize the effect of background polymerization. And so from other experiments, we see that around 10C, we can inhibit that. And so that's what we'll utilize for our application here. And then as a proof of concept to check the impregnation, we use the traditional resin fiber infiltration or pre-pregging scheme here, where we take fibers within carbon fibers on a fiber bobbin and actually align them through these tensioners, and then heat that fiber up as well in order to remove residual solvent as well as moisture. And then that's taken over the resin bath of the DCPD monomer that I'd showed previously. And then that's wound up and taken up on a drum. And so we've seen that this does in fact result in a composite that we can use in order to print. And so that really drives the development of the printer head that we're going to be using on our motion gantry. So we're going to translate the lessons learned that we did on the previous study in order to facilitate the impregnation, consolidation, as well as the deposition of the material. And so we take this and we miniaturize it and put it on a printer head. And so we have the carbon fiber inlet, similar to where the bobbin was. The matrix is being pumped into the head itself, and then there's a feed traction on the bottom that acts similar to the take-up drum in order to facilitate further impregnation of the material. And then that's printed on a heated print bed. And so taking a look at the inside of this, you can see the tensioners as well as the matrix roller, which is the main component here, where we have a radially aligned infusion ports. So the resin gets pumped in, and then that gets infiltrated into the carbon fiber tip. And so some preliminary work towards the printing of this, you'll see on the video in the upper right-hand corner, the fiber itself is held down initially, and then that material is then deposited onto a heated substrate, which then subsequently cures. And once that's fully cured, you'll see that we get properties around a volume fraction of about 30% with a moderate degree of cure of 85% and a TG of around 130. And taking an optical micrograph of this, taking an optical micrograph of the cross-section of this composite, you can see that there's insufficient voids in here, which is great, which is good news. And then we also have a good degree of impregnation as well. And so that was a simple demonstration of the technology. However, at the culmination of this project, we'll aim towards providing material manufacturing and integrated functionality. And so with 3D printing as well as composites, there's a large space that needs to be met. And so with that, I'll just look towards the summary as well as the future work towards this. So we're currently developing and demonstrated a method for 3D printing of these continuous fiber reinforced composites. And then we've developed a method for designing the structures taken from biology in order to get towards a functional performance. And the future work for this is integrating all of our subsystems together in order to fully actualize the 3D printing of bio-inspired structures. And with that, acknowledgements, I'd like to thank professors Scott White, as well as my advisors for this particular fellowship, Professor Nancy Sotos, Professor Philippe Gubel, Professor Sam Taufik, and Professor Mariana Kirsch, and then a lot of postdocs, as well as other graduate students and undergrads that played a hefty part in this, and my funding sources at the Air Force Office of Scientific Research, the National Science Foundation, and DARPA. So, any questions?
Thank you very much, Neil, and thank you for respecting the time as well, because it's kind of hard to focus it on in 30 minutes. Any question from anybody in the audience?
I have one.
Yeah.
Yes.
Hi, Neil. I'm so excited about this project.
Agreed.
Is it possible to print like plates and rods? Do you know what I
Yes.
mean?
Yeah, so one of the
Yeah, I noticed this. Yeah, the extruded.
so these so we could make cellular type structures given the modality that's given from the curing method that we're using. You can print freeform structures as well. So this allows us to print rods and then print plates on top of that rod if we really wanted to. So given the lightweight structure of the material.
I think it's going to be really exciting to compare a couple of different architectures and bones that have different functions, right? Like in compression versus bending, and see, if you can mimic that. That would be really exciting.
Yeah, I agree.
Okay, excited to see it. Thanks.
Yep.
Any other question in the audience? I have one quick question for you, Neil.
Yeah.
Is it important to have like kind of tension in the printed continuous fiber? Because I can see that you have tension inside the system, but once it's on the bed, is there still some tension?
Yeah, so the proof of concept that we gave here, we
Oh,
had
there's
to apply
something.
initial tension at the beginning in order to facilitate the extrusion of the toe. But
I got it.
given that we are using rollers for further facilitation, we're going to try to alleviate the need for that tension, and then act to also compact everything. So.
Okay.
Yep.
Great, thank you. We have time for one other question. Nobody else? All right, then we can move on to the third speaker today. So our third speaker is like Moitreya Chatterjee. I hope I'm saying that correctly. He's a PhD student in electrical and computer engineering and working with Prof. Narendra Ahuja. And his research encompasses computer vision and multimodal machine learning. And today he's going to talk about these two topics and towards an application in audio conditional visual synthesis. So Moitreya, the floor is yours now.
Thank you so much, Julie, for the introduction. So let me quickly share my screen and see if that works. Okay. So can everybody see my screen?
Yes.
Okay, sounds good. All right, so let me get started. So hi everyone, I'm Moitreya. I'm a PhD student in the EC department under Professor Narendra Ahuja. And today I'm happy to present our work titled Sound to Sight, Generating Visual Dynamics from Sound and Context. And this was a joint work in collaboration with Anup over at Mitsubishi and Professor Narendra Ahuja. A preliminary version of this work was presented at ECCB last year, and today I'm presenting sort of an adaptation from that. So let's take a look at the video. So we just saw the initial part of this video where we saw a person walking down the stairs, but then the later part of the video got occluded, but then we still heard the audio stream wherein we saw a person sort of where we heard a loud thud at the end, right? Now, this is a very real world, it simulates the real world quite accurately wherein, due to any number of situations such as occlusion or corruption of the visual stream, you might have a situation where you only have access to a few frames of the video, but then you don't have access to the entire video stream. However, you might still have access to the audio stream that accompanies the video, right? So what we want to do is to be able to reason over these sort of missing visual streams, and in particular, this is the setup that we have, where we are given a few frames and we have access to the entire audio stream, and what we want to do is extrapolate the frames into the future, right? So that they are consistent with the audio that has been observed. And this is actually very important from the perspective of creating truly AI systems. Because you want to be able to reason across multiple modalities, but then this also permits, compression, for example, where you can basically not store the frames that you can reason over, and then you save on data that way. Right? So yeah. So to be more specific, we address this problem of future frame generation from audio conditioned on the visual pass. And to the best of our knowledge, this is the first work in the deep learning domain that addresses this task. And towards that end, we propose a sound to sight, a model called sound to sight, which is basically a variational autoencoder coupled with the generative adversarial network setting to address this task. And we show state-of-the-art results on multiple data sets. So here's an overview of our model. And I think Michael earlier on in the day gave a primer on what generative adversarial networks are. Our model is also adapted from those GAN type architectures, wherein we have a generator which recursively generates one frame at a time, having accepted the audio stream A1 to T and the visual frames that have been generated so far. Along with it is what is known as the discriminator architecture, which basically is responsible for classifying a stack of frames as being belonging to a real or a synthetic form, right? And as is popular in the GAN literature or the GAN area, these two components are trained adversarially, where the generator is supposed to try to fool the discriminator, while the discriminator tries to do as good of a job at classification as it can. And once this is trained to convergence, basically what's known as the Nash equilibrium, what you would get is the generator would be in a position to generate very realistic looking frames. So we leverage that architecture, right? And really the key generation workhorse of our model is the generator component. So let's delve a little deeper into that. So here's an overview of our generator architecture. The key component of our model is this prediction network, right? What it does is it takes as input this frame x hat t minus 1, which is the last frame that has been generated. And then it basically, it also accepts as input a stochasticity vector zt. And with this pair of inputs, it's able to generate the current frame, which is x hat t, right? However, okay, so the reason we add the stochasticity is basically because a direct forward estimate from x hat t to x hat t minus 1 to x hat t doesn't really model the stochasticity or the uncertainty that exists in the real world. So that's why we incorporate this ZT vector, which is a stochasticity component, which allows for some perturbation beyond what the model expects. Right. Now, this ZT in the standard Kalman filter type setting might be estimated from known distributions. However, what we do is we learn the distribution from which the ZT is estimated. In particular, we model it with the Gaussian, but then its parameters, the parameters of this Gaussian are learned from data. And what we do is we basically learn two models, one called the posterior, the other the prior. Note that the posterior takes as input the frames x1 to xt during training, and wherein x at t is what we want to generate, meaning an approximation of xt is what we want to generate, and the audio stream is fed as input as well. And along with that is a prior network, which is taking as input the frames x1 to t-1, and along with it is supplied the audio stream as well, a1 to t. So with these respective sets of inputs, the posterior estimates a Gaussian distribution parameters by psi, while the prior estimates another Gaussian distribution parameters by phi, right? And what we do is during training, we try to match these two distributions as closely as possible to each other, so that post the training, the KL loss goes to 0, and then you could basically get rid of the posterior and operate only with the prior. Now, what that does is that frees up the model's dependency on this frame x at t, and you can only operate with the frames x1 to t-1, and of course the audio stream is available to you. And with that, you can just estimate this distribution, this Gaussian distribution, sample the ZT from there, feed it to the prediction network, and that can then proceed to generate x at t. In terms of the generator, in terms of the loss, we have this generator loss that we are actually that is used to train this generator, wherein this mean squared error term basically tries to match the predicted frame with the ground truth one, and then there's the scale loss term which tries to match the posterior and the prior distributions. All right, so here's the experimental setup. So we conduct experiments on 3 datasets. The first one is a adaptation of the moving MS dataset, where a digit is moving in a block, and then occasionally at random locations, a box appears, such as the one shown in white here. And the digit is colliding either with the edges of the box or with the block. And when it collides, it randomly changes the direction of motion, right? Randomly. Along with it, there's an audio that accompanies the digit. And every digit 0 to 9 has its own associated frequency with it. That frequency also gets perturbed when there is a bounce that happens. We also show results on real-world data, such as the person drumming a drum kit here. And here the goal is that you only have access to a few frames, and then again you have to extrapolate the frames in the future. But then you have access to the entire, the drum audio that I use. So how well of a generation can you do with that setup? And then the final data set that we report on is a data set that we crawl from YouTube of painters painting on a canvas. Of course, again, the setup is similar. You have access to only a few initial frames, and then you can extrapolate the frames into the future. But then you have the accompanying audio available too. And the audio here emanates out of the paintbrush scratching against the canvas. And that's really the only audio cue that you can get. So here's the quantification of the results. And well, the performance here, metric used here is the peak signal to noise ratio. And what we see is that our model, highlighted by the red block, outperforms some of the baselines that we came up with. And in the paper, there are other metrics that we report on as well, and I'll just refer you to that if you're more interested in this. Here's some qualitative results. So we see that our method is able to do a much more accurate job of generating the video frames compared to any of these baselines that are shown to the right. Either they don't model the digit well, or they just generate some blurry generations that are not good enough. On the painting dataset, we see that our method is able to generate somewhat of a more accurate generation that mimics the ground truth more closely as compared to the other base methods, which either show some artifact or either generate blurry generations here or basically show no motion in this case. So yeah, and also added to that, because our model is stochastic in nature, it permits diverse generation, and so we see the diverse generation by our model. So there are more results that we point to in the paper, and I'll refer you to that in case you're interested more. And here's a link to our project page, which has all the code, data, more details, the link to the paper. So thank you for your patience, and I'm happy to take questions.
Great, thank you, Moitreya. Great talk. Any question in the audience?
Maybe I'll just take you to the model slide.
So I have a very general question.
Sure.
Based on what I understand and based on what you present on what you presented, I'll take an example just so that you can understand my question. Imagine you have a 10-second video and you have like a 25-second audio. Are you able to predict the 15-second left that is like even more than what you have in terms of data? Like what are the ratio of like the known versus the unknown that you can predict basically in terms of time?
Yeah, so that's a great question. So actually, I'll just show you here. Yeah, so if you actually look at this, so the M3SO, which is the moving and mist data set that we report on, you see that the prediction actually starts from frame 31. So you've only seen the first 30 frames, whereas here we're reporting numbers up until the 70th frame. So like you're extrapolating on the next 40 frames, basically. So I think the frame rate for this was, I think, 10 frames per second. So you could say that, you're looking at the first 3 seconds of the video, but then predicting the next 4 seconds of the video. Our model can extrapolate even further down, but then I think the quality degrades a bit. But yeah, I think it's still feasible. Yeah.
And still based on that question, does it help to have a better resolution, or is it not related to resolution at all? Resolution of the images, the spatial resolution of the images.
Got it. We have varying resolutions that we report on. For example, this moving and mist is reported on 48 by 48 videos. The real world videos are 64 by 64. So it's true that it's perhaps not super high resolution at this point in time, because once you get into the very high resolution stuff, you have to take care of the details of the background, things that are not necessarily related to the key event of the motion that's happening. But yeah, so far we're just dealing with 64 by 64 videos for the real world case.
Okay, thank you. Any other question? We have like 2 to 3 minutes left, so feel free to ask more questions. That's your last call. Well, if it is, then I'm gonna thank you all for attending today this seminar. I hope you enjoyed it. Like I enjoyed it, so that's great at least. And it's great because especially like the speakers took like they really showed that how we can do interdisciplinary research in Beckman. So I think that was a great session today. So have a nice afternoon. It's the evening for me here, but have a nice afternoon, everybody. And the next Beckman graduate student seminar is on October 13. So feel free to join again.
Thanks, everyone. Bye.